something that's actually alive, not just organ systems or chemistry. We're now talking about the smallest units that are actually alive in an organism. And since we're talking about humans, we're talking about human cells. But cells belong to all organisms, and there is a concept in biology known as the cell theory, which can be stated in several different ways, but this is a good one. Cell is the structural and functional unit of life. Everything that happens in an organism, everything that you can see in an organism, is because of the activities of single cells. The entire organism is going to depend on the combined activities of all those cells, particularly in multicellular organisms. There are single-celled organisms, but humans obviously are multicellular. Remember, structure and function are have complementarity. The biochemical function of cells is going to dictate the cell, the shape of specific subcellular structures, and most importantly, continuity of life has a cellular basis, so cells only arise from pre-existing cells. We don't get spontaneous generation. There are at least 250 different types of cells in a human, and that's probably a low estimate, conservative estimate. The types differ in size, shape, some of their subcellular components, but they're all related to the specific functions that those cells have. So we've got everything from fibroblasts to erythrocytes, macrophages, cells of reproduction, nerve cells, many different kinds of cells. The generalized cell, what we can look at as a cell that doesn't really exist, but it's got most of the components that we'll find in other cells, would start with three basic parts. First, the plasma membrane around the outside, flexible outer boundary. Probably heard of a phospholipid bilayer before. Beneath that would be the cytoplasm, where we would have all kinds of organelles that we'll talk more details about, especially in this chapter. And then within the cytoplasm, usually around the center of the cell, there is an organelle called the nucleus, which contains the DNA and is the control center of the cell because it determines which set of DNA, which genes are going to be used by that particular cell at that particular time. So here is our nucleus, and structure inside it called the nucleolus, mostly DNA in the form of chromatin in most cells. Then what we call cytoplasm is actually filled with all kinds of things: endoplasmic reticulum, rough and smooth, mitochondria, Golgi and vesicles, all kinds of things that we'll talk about in detail, but in general called cytoplasm, and then separated from the rest of the world by the plasma membrane. There are also things outside the cell that are produced by cells. There would be in general called extracellular materials. There are classes of extracellular materials that would include fluids. The body fluids, we'll just use its general term sometimes. The interstitial fluid is the fluid that's right around the individual cells. Cells are bathed in this fluid. Blood plasma would be another example. That would be the fluid portion of the blood. Cerebral spinal fluid we'll talk about when we talk about the nervous system, particularly the central nervous system. We also have secretions from cells that would include saliva and mucus. And then we'll talk about connective tissues next week when we talk about tissues, but connective tissues secrete things called extracellular matrix. There are several different kinds of them, but they're all substances that hold cells together, give structure to the body. So let's start with the plasma membrane. The plasma membrane is an active barrier that separates the intercellular fluid, which really you can think of as cytoplasm, from the extracellular fluid, the interstitial fluid we just mentioned. It has a very dynamic role in controlling cell activity because it ultimately determines what goes in or leaves the cell. It is a a selectively permeable membrane. It is sometimes referred to as the cell membrane. In fact, it's often referred to as a cell membrane, but you should be careful using that term because any membrane of a cell is technically a cell membrane. Plasma membrane is made of membrane lipids that form the flexible lipid bilayer, which I hope you're familiar with. They are filled with proteins. In biology, we talk about transport proteins, but there are many other types of proteins that are there. They are all floating in that phospholipid bilayer. So it was remember the fluid mosaic model is used to describe the plasma membrane. They are moving around in patterns. They interact with each other. There are sugars, carbohydrates on the surface of the protein that form a structure known as the glycocalyx. And there are many other structures that help hold the cells together, one cell to another, called cell junctions. So here's our plasma membrane, a phospholipid bilayer. Remember the phospho groups and the lipid tails on the phospholipid organize themselves so that the phospho groups are inside and outside. The lipid tails are in the middle. We have transmembrane proteins. We have proteins that exist on just one side or the other, connections to the cytoplasm, and carbohydrates forming the glycocalyx. And those are all shown here. Phospholipid, cholesterol within the lipids themselves, proteins, and carbohydrates.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Okay, there we go. So that lipid portion of the membrane is made up of about 75% phospholipids, which we've talked about before. They just mentioned in the video. They have a phosphate head, which is polar. It has a charge on it, so it likes to be in water. It's hydrophilic. And the other side of that molecule, so it's all within the same molecule, is a fatty acid tail, which is nonpolar. And for that reason, it's hydrophobic, water-hating or water-fearing. Another 5% of that membrane lipid is glycolipids, and those are lipids with a sugar attached to the outside surface, so that would contribute to the glycocalyx. And then in the membrane, there is about 20% cholesterol, which increases the membrane stability, so it makes it a better membrane. It also makes it a more fluid membrane, so that those phospholipids and the things in them can move around more freely in our membrane, or fluid mosaic model of the membrane. So here, just again, is the polar head, non-polar tail of a fatty acid. When you put those into a water environment, they organize themselves into structures, small structures we call micelles, which you can expand to make an image of the cell itself. The polar heads, the polar heads face inside and outside. The lipid tails form a little barrier in between. Proteins within the membrane allow for communication with the environment. Many of them are transport proteins. They make up about half the mass of the plasma membrane, and they all have specialized membrane functions. Many of them float freely in our fluid mosaic model, but there are some that are tethered to intracellular structures. So we can divide proteins in the membrane into two types: the integral proteins, which pass through the membrane, and the peripheral proteins, which are found on only one side of the membrane. So those integral proteins are firmly inserted into the membrane. They are mostly transmembrane proteins. They span the membrane. They go from inside to outside the cell or outside to in, depending on your orientation. So that means they have to have both hydrophobic and hydrophilic regions. The hydrophobic regions interact with the membrane itself, the lipid tails of the membrane, while the hydrophilic areas will interact with the extracellular and intracellular portions of the membrane. They're hydrophilic, they interact with water. A lot of these will function as transport proteins. They may be channels, which are kind of like doors that are always open, or they may be carriers that pick up something on one side and move to another. A lot of them are enzymes. Many of them are receptors. We will talk about a lot of them through the course of the year, though we won't really discuss their integral membrane structure throughout the course of the year. The peripheral proteins are loosely attached to integral proteins. They include filaments on the intracellular surface for plasma membrane support. Again, they have very different functions. They could be enzymes, could be motor proteins used to change the shape of the cell during division or muscle contraction. They could be used for cell-cell connections. So they perform many different tasks, transport being the first one. Transport proteins allow things to move from one side of the membrane to the other. Some of them use ATP as an energy source, others do not.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood.
I really hate that video. Hemoglobin is a transport protein. It's not a membrane protein. It does carry oxygen, but doesn't help oxygen cross plasma membranes. But it is a transport protein. Receptors are going to bind to chemical messengers and cause signals to occur within the cell.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
A lot of them have enzyme activity. Even receptors, when they bind, they will have an enzyme activity on the other side and catalyze a response.
Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
Then we have cell-cell recognition. A lot of glycoproteins, the sugars attached to there provide identification markers, and many cells look for those identification markers, particularly those cells of the immune system, to protect us from disease. Okay, let's see if it'll start. Nope. Try it one more time.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
Okay, and then, we have attachment to the cytoskeleton or the extracellular matrix, depending on whether it's internal or external. That's going to help maintain cell shape, fixes it in certain locations, and has a role in cell movement when movement is part of the cell's job. We also have connections that are for cell-cell joining. They're hooked together with cell adhesion molecules, which we'll talk about later on in section 6, I believe. They provide temporary binding sites and guide migration. So that glycocalyx is sugars or carbohydrates that are sticking out on the cell surface. Some of them are attached to lipids, some of them attached to proteins, so they'd be glycolipids or glycoproteins. Every cell has its own pattern. You can think of it as a sugar coating. Much of it serves as a biological marker for cell-to-cell recognition. And a big part of that allows our immune system to recognize what is ours and what is not. So it differentiates between self and non-self. It allows cells that are self to remain, but things that are non-self, it tries to get rid of. We'll talk a lot about that when we talk about the immune system, but it's one thing you may know, may be aware of, is blood types. Your blood types, your A and B blood types, the ABO system. A and B are both carbohydrates expressed on red blood cells. Cancer cells can sometimes change their glycocalyx, and that can either evade immune response or, with any hope at all, help to speed up an immune response. Some cells are free. They're not bound to other cells. The blood cells, for example, sperm cells. Most cells, though, are bound together in the form of tissues which form organs, and there are three different major ways they can be held together: tight junctions, desmosomes, and gap junctions. In a tight junction, the integral membrane proteins fuse and make an impermeable junction that encircles the whole cell. Very good for preventing fluid and most molecules from moving in between cells, and you should ask yourself where those might be useful in the body. We'll go over that when we talk about tissues next week. So here we have a tight junction, tight junctions. The second one are desmosomes. These are more rivet-like cell junctions formed where linker proteins that are known as cadhedrans of neighboring cells kind of interlock like a zipper. These linker proteins are anchored through button-like areas, so it's not the entire plasma membrane, but regions of the plasma membrane called plaques. Keratin filaments connect the plaques intracellularly for added anchoring and strength. So these allow for some give between the cells, which helps to reduce the possibility of a tearing of this type of membrane when it's under tension. So there is an example. And gap junctions are transmembrane proteins called connexons that form tunnels in between the cytoplasm, directly connecting the cytoplasm of two adjacent cells. And these can be used to spread ions, simple sugars, and other molecules between cells without having them leave the cell. Also allows for electrical signals to go quickly from one cell to another. We're going to see these in cardiac cells, where we're going to call them intercalated discs, and they are also present in smooth muscle cells, which we'll talk about when we talk about smooth muscle cell contractions. All right, we will pause it there, and the next thing we'll cover is passive and active transport.